Hi, good morning, one and all. Today we can discuss about a very important topic, that is urine formation. See, last class I already said the each kidney they having a 1 millions of nephrons is present. Nephron is the structural and functional unit of kidney. The nephrons is two types. One is the cortical nephron, another one is the juxtamedullary nephron. Now I already said in kidney this outer part is called as a cortex and inner part is called as a medulla. This is a conical shaped structure. This conical shaped structure is called as a renal pyramids, and this one is the renal papillae. What is differentiation between the cortical nephron and juxtamedullary nephron means? Suppose, see, you have to think this one is the kidney. The outer part, that one is the cortex, and inner part, that one is called as a medulla. You know, very familiar, the nephron. See, the nephron having, what are the parts is present? They having a glomerulus, they having a Bowman's capsule, they having a proximal convoluted tubule, they having a loop of Henle, distal convoluted tubule, and collecting duct. See, collecting duct, it is not in the part of nephron. You have to keep in mind. That's why, see, what are the parts is present in nephron? Glomerulus, Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule. Now this part is called as a cortex and this part is the medulla. This is the glomerulus, this is Bowman's capsule, this is PCT, this is loop of Henle, and this is DCT. This is totally a straight collecting duct. What is differentiation between cortical nephron and juxtamedullary nephron means? Here glomerulus, Bowman's capsule, PCT, DCT is present in the cortex region. Loop of Henle present in the medullary region. Collecting duct both present in cortex region and medullary region. You have to remember that regions. What is differentiation between cortical nephron and juxtamedullary nephron means? In cortical nephron, loop of Henle is very short, but in juxtamedullary nephron, loop of Henle is very long, present deep in the medulla or present deep in the renal pyramid, but this is present in the apex of the renal pyramid. That is the main differentiation. Here, now we can go for the urine formation. For urine formation, mainly we need three steps. What are the three steps? The first step is the glomerular filtration. The second step is the tubular reabsorption. The third step is the tubular secretion. Correct statements mean tubular selective reabsorption, tubular selective secretion is the correct one. Now, here, according to the urine formation, first we can go for the structure of a nephron. See, from renal artery, this artery is called as a efferent artery. Effa means towards. When this afferent arteriole, this is Bowman's capsule, they having a two layers. When this afferent arteriole enter into the Bowman's capsule, this afferent arteriole is divided into bunch of or tuft of blood capillaries. This tuft of blood capillaries is called as a glomerulus. That's why, what is glomerulus? Glomerulus is a tuft of blood capillaries formed from the afferent arteriole. When this glomerulus, this blood vessels can leaves the Bowman's capsule, that is called as a efferent arteriole. Effer means away, affer means towards. Now the glomerulus is formed. This glomerulus is surrounded by a double layer structure that is called as a Bowman's capsule. In this Bowman's capsule, outer layer is called, this is the outer one, outer layer is called as a parietal layer, inner layer is called as a visceral layer. Between outer parietal layer and inner visceral layer, they having a space. This space is called as a capsular space. Okay, now see, for now here the blood is enter. Which type of blood? The blood with the waste materials enter. Here the blood filtration is takes place. That's why that we can call as a glomerular filtration. When the blood is filtration here, filtration is takes place by the three membranes. One is endothelium of glomerulus. It means what I said, glomerulus is the blood capillaries. In that blood capillaries, the inner layer is called as a endothelium of blood capillaries, and this is inner layer, that is called as a visceral layer. This visceral layer, they having a podocytes. They can arrange like a intrinsical manner. It means they having a pore, there is no gap. They having a pore, there is no gap. Like that is called as a intrinsic manner, that is called as a podocytes. That's why here visceral layer of podocytes, between glomerulus and between this Bowman's capsule of inner layer, they having a membrane. That membrane is called as a basement membrane. That's why based upon these three layers only filtration is takes place. That's why first the blood is touches the endothelium of the glomerulus, next to the basement membrane, next to the Bowman's capsule of a visceral layer, and they having a podocytes. Here when this blood is enter here, the filtration is takes place. How the filtration is takes place we can discuss later. On average 100 to 120 ml of blood is filtered by the kidney per minute. Basically for minute, how many heartbeats is takes place? 72 heartbeats. Each heartbeat they having a how much amount of blood is pumped? 70 ml. Approximately you can get 5000 ml. Out of 5000 ml, how much amount of blood is filtered? 1100 to 1200 ml. Can I say approximately 1/5 of the ventricles blood is pumped? Is there or not? Now in this, how much filtrates is formed? That is called as a glomerular filtration rate. Here the glomerular filtration rate per minute, how much amount of blood we have to perfectly filtrate? It means 1100 to 1200 ml comes here, but filtration is takes place how much ml? 125 ml per minute. That's why here 125 ml per minute means per day, it means 24 hours into 60 minutes you can get 180 liters per day is the glomerular filtration rate. Now we can go for the structures. I already said this is the afferent arteriole, and this one is the glomerulus, and this one is the efferent arteriole. Now here this is PCT, this is loop of Fenley, and this one is the DCT. This is called as the initial collecting ducts. All the initial collecting duct will be combined, they can form a straight collecting duct. Now see, when this efferent arteriole, when it can leaves the Bowman's capsule, now this blood capillaries is called as a peritubular capillaries. When this peritubular capillaries parallelly wrapped around the loop of Henle. Parallelly wrapped around the loop of Henle. Now this is called as a vasa recta. That's why vasa recta is formed around the loop of Henle. And another thing, this peritubular capillary is densely packed around the DCT, that is called as a macula densa. Or as per NCERT, they gave the afferent arterioles is tightly wrapped around the DCT, that is called as a macula densa. Okay. It means NCERT statements they gave why afferent arteriole means. Before I said peritubular capillaries is wrapped around the DCT, that is called as a macula densa. I said peritubular capillaries, where it is arises? It is arises from the efferent arteriole. Where the efferent arteriole arises? From the glomerulus. Where the glomerulus arises? From the afferent arteriole. That's why here they can give as a afferent arteriole is wrapped around the DCT, that is called as a macula densa. This is the structure according to the nephron. Now we can go for the urine formation. In urine formation, first what step I said? Glomerular filtration. The second step is the tubular reabsorption. Third step is the tubular secretion. Glomerular filtration can be takes place in the Malphizian tubules. What is Malphizian tubules? Glomerulus and Bowman's capsule combinedly called as a Malphizian tubules. Suppose in this glomerular filtration, the blood is filtrated. When the blood is filtrated, compulsory they needed three types of pressures, very important. One is glomerular hydrostatic pressure. Second one is the colloidal osmotic pressure. Third one is the capsular hydrostatic pressure. First, when the blood is enter from the afferent arterioles to glomerulus. See, I already said in blood mostly they can consist of what? Plasma. In plasma mostly they can consist of a water. Through water it can exert a pressure like this. It means fall down like this. When it is exert a pressure like this, that pressure is called as a glomerular hydrostatic pressure. How much pressure it obtained here? 60 mm of Hg pressure. Suppose when this pressure like this, compulsory opposite force also can be exerted. What are the opposite force means? One is colloidal osmotic pressure, another one is the capsular hydrostatic pressure. Why? Because when forcefully blood is enter into here, some large plasma proteins is there. This large plasma proteins opposite it can exert a pressure like this. When oppositely exert a pressure by large plasma proteins, that pressure is called as a colloidal osmotic pressure. That is how much mg? 32 mm Hg pressure. Now third one, when blood is enter like this, between the space is called as a capsular space. This capsular space having a fluid, that is called as a capsular fluid. This capsular fluid also oppositely it can exert the pressure like this. That is called as a capsular hydrostatic pressure. That is how much mg? 18 mm Hg pressure. Now the net filtration pressure is, one force is coming like this, two forces oppositely exert like this. This force is what? Glomerular hydrostatic pressure. That is how much? 60 mm Hg. And opposite force are colloidal osmotic pressure and capsular hydrostatic pressure. 60 minus 32 plus 18. 60 minus 50, that is what? 10 mm Hg pressure. That's why the net filtration pressure we have to maintain 10 mm Hg pressure. When this 10 mm Hg pressure we have to maintain here, the blood is filtrated. Now filtrates is formed. Okay, now this filtrates is called as a primary urine. Okay, now the remaining of tubular reabsorption and tubular secretion we can discuss in the next class. Thank you.